Hello again and welcome to Olympian. This is Aditya Mandal and today we are going to be talking about DNA replication. Now this is essentially a continuation of my previous lecture in which I talked about DNA or deoxyribonucleic acid in general, and today let's delve into the details of a particular process that DNA undergoes, which is replication. All right, and let's begin today's lecture. Now, before we begin, let me just ask you one thing. How do you think DNA replicates? Like, how does it look like? How does DNA replicate? You might not be getting the question, but once I draw some diagrams, you'll probably understand. How does DNA replicate? Now, let me give you some options. Okay, so let's say the first option is I have a DNA that's like this. I have a DNA that's like this. Let's just assume that this is a double helical structure. I'm not drawing the helices in quotes, the helices per se. I'm just kind of imagine that the straight lines are the rungs of the spiral staircase, right? And now the DNA goes to something like this. This is the original DNA, and the one I'm drawing in yellow, this is the child DNA, right? The daughter DNA, what we call. Okay, what do you think? Is it like this? And then finally, when the entirety of the DNA is replicated, there's the original left behind like this, and the daughter like this, right? So, what do you think? Is this the way it happens? Let's see. Let's go further. Let's go deeper into the topic. And let me give you another option, option number two, which is, let's say this is my parental DNA. And have you ever seen budding in Hydra, in organisms like Hydra? What happens? You get the original one like this, and a kind of tiny version of the original coming out of the side like that. So, what do you think? Does it happen this way? Does it happen this way? What do you think? Let's see. And this finally goes to two pieces of DNA like this, right? I hope you can distinguish between the red and the yellow because that's very important. Let me now give you the third option. I have a DNA like this, and I make the arrow, and there's the Y, and you notice that both the arms of the Y are in the same color, that is red, means it's both parental, and the strands of the new DNA, they are generated like this. The yellow is the new DNA. What do you think? Is this what happens? Is this what happens? Maybe all of the three are possible, doesn't it? Isn't it? All of the three seem possible, but how do we understand which one is the process that actually happens, right? Which one is the process that actually happens? And you may already know which one is the process that actually happens, but that you probably just know that as a fact now, right? You don't know all the things that went in. Oh, sorry. I should change it to area eraser. Yeah, you probably know it as a fact, right? And by knowing this sort of thing as a fact, we fail to appreciate the amount of research, the amount of what do I say, the intelligent deductions, intelligent guesses, because we can't really see the DNA, right? We can't see how it is replicating. So it's, after all, it's just a deduction, right? So it's not a very easy task to reduce which one of these processes actually takes place, because you see the three, they are all equally, not exactly equally likely, but they are all possible, right? They are all possible. So how do you know, before this is before we have a very detailed study of DNA, this is before we have a field of science that is entirely dedicated to DNA. This is before we have even this is before we have sequenced any organism's DNA. This is before we have seen the DNA. All right. So how do we understand which of these processes is actually taking place? Take a bow, Messrs. Messelson and Stahl. Messelson and Stahl, right? These are the people who actually performed the path breaking experiment that lets us know which of the aforementioned processes actually takes place when DNA replicates, right? And I'm going to be talking about this experiment. Okay, so in Messelson and Stahl experiment, they, as is quite evident, they were geniuses, right? So what they did, you know that DNA comprises of nitrogenous bases, right? And if you've watched my previous video, I have delved into the structures and everything about the nitrogenous bases, the purines and the pyrimidines in detail. And I would recommend watching that video because the basics that I have expounded on in that video are required to understand this video, right? But let me just recap in brief in a sort of way, right? So the kinds of bases, the nitrogenous bases that are present in DNA can be divided into purines and pyrimidines, right? Purines include uh, adenine and guanine, and the pyrimidines include thymine and cytosine. And adenine binds to thymine via 2 hydrogen bonds, and guanine binds to cytosine via 3 hydrogen bonds, right? So nitrogenous bases, what does the name itself contain? Nitrogenous, nitrogen, right? Nitrogen. Okay, so nitrogen is present in all DNA, right? Nitrogen is an essential component of all DNA, and nitrogen is usually represented as 14N7, right? 7 is the atomic number, 14 is the mass number or nucleon number. To be if I go further to the basics of this, if you don't understand this notation, then let me break it down even further for you. 7 is the number of protons present in the nucleus of the nitrogen atom, and 14 is the total number of protons as well as neutrons, which we collectively call as nucleons present in the nucleus of nitrogen atom, right? So 14 7n, right? So if 14 is the total number of protons and neutrons and 7 is the number of protons, and what is the number of neutrons? 14 minus 7, right? 14 minus 7, which is 7. Okay, so this contains 7 protons and 7 neutrons, and this is the most abundant isotope, which usually the nitrogenous bases in DNA, they usually contain this type of nitrogen because it is the most abundant isotope. So the nitrogenous bases contain this type of nitrogen, yeah, right? And what is isotope? Do you know what is isotope? Isotope is two isotopes are two atoms of the same element which contain the same number of protons but different number of neutrons. So what do you think changes? It's a mass number, right? Because the number of protons is the same, so the atomic number, the subscript, must remain the same, right? And 147N, as I told you, is the most abundant isotope, and usually nitrogenous bases are composed of this type of nitrogen only, right? Now, there is another isotope, 157N. Oh my god, what is this? What is this? This is an isotope that is less abundant, but what do you think it contains extra? It contains 1 neutron extra, right? 15 minus 7 is 8. So 8 neutrons is what it contains, right? 157N is 15 is the mass number, 7 is the proton number, the atomic number. So 8 is the number of neutrons, and this is called heavy nitrogen, right? Why is it called heavy? Because quite simply its mass number is more, right? Its mass is more compared to nitrogen. It contains 1 neutron extra. So if you consider a mole of this nitrogen, it will be heavier than a mole of that nitrogen, right? If you can consider a mole of 157N, it will weigh 15 grams if you know Avogadro's law. And a mole of 147N, it will weigh 14 grams, right? So this is heavy nitrogen, which is a less abundant isotope, right? Heavy nitrogen. Now you must be like wondering, why is this coming into the picture here? What good does that do us? What do we need this for? This is important because people like it takes a genius to understand the importance of something which seems as trivial as this, right? And Messelson and Stahl were geniuses, and what they did is they grew DNA. They grew DNA for several generations. They grew DNA for several generations in a culture of 15 NH4Cl. What is NH4Cl? Ammonium chloride. And I'm not writing the atomic number anymore because it's quite evident. But 15 NH4Cl signifies that the nitrogen used in the culture is heavy nitrogen, right? They prepared a special culture of 15 NH4Cl and they grew the DNA for several generations in that, right? They grew the DNA. Now, what happens when you grow DNA for several generations in 15 NH4Cl? Well, when DNA replicates, it the biosynthesis of the nitrogenous bases, it takes place from the medium itself, right? Now the nitrogen present in the medium is the nitrogen that gets incorporated into the nitrogenous bases, right? So the nitrogen present in the medium is 15 NH4, is 15 nitrogen, right? The heavy nitrogen. So all the nitrogenous bases, all the nitrogen contained in all the nitrogenous bases will be heavy nitrogen, because the biosynthesis of those nitrogenous bases is taking place using the nitrogen contained in the medium, right? So all the entirety of the DNA will be heavy DNA. We call it heavy DNA because all the nitrogenous bases are comprised of heavy nitrogen, and therefore we call it heavy DNA, right? Both the strands of DNA, it's heavy DNA, right? 15NH4Cl, heavy DNA, right? Then they conducted a CSCl. They conducted a CSCl. CSCl is cesium chloride. It's a compound. It's a laboratory technique, density gradient centrifugation, right? CSCl density gradient. Now, what happens in centrifugation? In centrifugation, basically the the DNA or whatever substance is being centrifuged, it is taken in a special apparatus known as a centrifuge. A centrifuge. It's known as a centrifuge. CENTRIFUGE. It's known as a centrifuge, and it's rotated at very high speeds, right? It's rotated at very high speeds. And what happens? You can either use the concept of centrifugal force here, which I personally don't like because it's a pseudo force, but that's physics, and this is a biology channel. I won't delve too much into the physics here. But what happens is that the heavier particles or whatever you are centrifuging, they move towards the outside of the tube that is being rotated, and the lighter ones remain towards the center. Okay, the lighter ones remain towards the center, right? So they conducted a CSCl density gradient on the DNA. Now you realize, right? 15NH4Cl is composed of heavy nitrogen, so the DNA is heavy DNA. It will be heavier than normal DNA, right? So it will be a little further away towards the side of the tube, towards the edge of the tube, than the normal DNA if it is rotated in the centrifuge, right? I'll just remove the centrifuge. There's no business being here. So now the results of the CSCl density gradient. Let's say this is the I am rotating the tube, and let's say this is the mouth of the tube. This is the mouth of the tube, and this is the base of the tube, right? So most of the heavy DNA, it will be around here, right? It will be around here. Most of the heavy DNA will be around here, and it will be a single band, right? They conducted a CSCl density gradient in since all the DNA is heavy DNA, 15NH4Cl DNA, so all of it will accumulate in more or less the same place, right? In more or less exactly the same place. So it will form a single thick band comprising of 15NH4Cl DNA or heavy DNA. And let me just label it here. This is heavy DNA, right? This is heavy DNA. Okay, so it just forms a single band, and this is the CSCl density gradient, and this is the first step of the experiment, right? Okay, now let's move to step 2. they now transferred the heavy DNA, the heavy DNA to a medium of 14NH4Cl, right? 14N, that is the normal N, what we call normal nitrogen, and it's not heavy, it's the most abundant isotope of nitrogen, and they transferred this heavy DNA. Well and good, right? Very good, they transferred it. Now, after 19 minutes, 19 minutes has a significance which I'll be talking about later. In some books, you may also see this written as 20 minutes, but 19 minutes is actually more accurate. After 19 minutes, they conducted another CSCl density gradient. They conducted another CSCl DG, right? Another CSCl density gradient. Now what happened? Now what happened? Okay, let's say this blue line was the initial position where the heavy DNA was present. Okay, now all the DNA is here. All the DNA is here. That is bamboozling, right? All the DNA, it appears to have shifted, right? All the DNA is at an intermediate position, a position that is before the position of the heavy DNA, but not exactly to the mouth of the tube, right? It is at an intermediate position, and this is known as intermediate DNA. You will understand the nomenclature later. And they put another mark on the density gradient tube signifying where the intermediate DNA had accumulated, and they saw that it was more towards the mouth of the tube than the line of the heavy DNA, right? Than the line of the heavy DNA. Okay, now after another 19 minutes, after another 19 minutes, total 38 minutes, that is, okay, total 38 minutes, total 38 minutes, they conducted. Now, by conducted, I don't mean they took the whole tube of DNA and they put it in the CSCl. They just took small samples, right? So that the further steps in the experiment could be conducted. It's a laboratory procedure, right? We don't take the entire thing and we dump it in the tube and we rotate the tube. This is a very strictly controlled procedure. We just take small samples and we do whatever we want with it, right? They conducted another CSCl density gradient, right? And now if I draw the tube, this is the tube. What was the result? Let's say this is the line of the heavy DNA, this is the line of the intermediate DNA. Now they saw that the line of the intermediate DNA was thinner, the line of the intermediate DNA was thinner, right? And there was another line. Oh my god, there was another line of the same thickness as the line of the intermediate DNA. What's happening? How is this possible? Like what is this procedure? I can't even understand what it implies. Now do you get it why this was such a path breaking experiment, why they are given the title of geniuses? Because it's not very easy to interpret the results of these experiments, right? If you don't know about what this experiment actually entails, what inferences can be drawn from this experiment, as a neophyte you probably can't glean anything from this experiment, right? So this is a line of intermediate DNA as before, intermediate DNA, and you see this line that's closest to the mouth of the tube. This is known as light DNA. You'll understand why later. But this is it's actually logical, right? Because it's has to be the lightest if it's towards the mouth of the tube because it's rotating, and centrifugally the heavier bits will move towards the outside and the lighter bits will remain towards the closer to the mouth of the tube, right? So this, logically speaking, the name light DNA is justified because it's the closest to the mouth of the tube, right? And you'll understand why biologically it is justified as well when I come to the inference of the experiment, right? The inference of the experiment. Okay, after another 19 minutes, after another 19 minutes, that is a total of 57 minutes, right? Total 57 minutes. They conducted another CSCL density gradient. CSCL density gradient, right? So now what did they see, right? Now what did they see? Let's say this is the line for heavy, this is the line for intermediate, this is the line for light, right? Now they noticed that the line of the light DNA was this thick, and the line of the intermediate DNA was very thin, this thin, right? Thinner than the previous one. And from measurements, they actually gauged that the line of the light DNA was 3 times thicker than the line of the intermediate DNA, right? It was 3 times thicker. That value has significance. So why it is 3 times thicker, it actually has significance, right? So this is, I'm not writing light and intermediate anymore because it's basically the same. But this is the intermediate DNA, the line of the intermediate DNA, and it's 3 times thinner than the line of the light DNA. How is that possible? We'll come to it in the inferences. For now, just trust me, right? For now, just trust me. Okay, I think this should be enough to move to the inferences of the experiment, right? And since I'm kind of a stickler, I'm kind of a pedantic, let's just write that in as well. No harm done. Intermediate DNA and light DNA. Now inferences. Inferences are the most important. If you just do an experiment, it's of no use, right? If you can't infer anything from it. And the correct word to use here is infer, not imply. Infer and imply are often used interchangeably, but that's wrong. The correct word to use here is inference. This, what can be inferred from this experiment. Imply can also be used. What does this experiment imply? But then the tone of the sentence, then the it becomes more active rather than passive, right? So inferences. So this experiment, drill this statement into your brain, right? This experiment proved semi-conservative replication of DNA. What is semi-conservative replication? Semi-conservative replication of DNA. Semi-conservative replication of DNA. What is semi-conservative replication? Let me go back. Option number 3 is semi-conservative replication. Okay, option number 3, this is semi-conservative replication. So you finally got the answer to the question which I had posed before you at the beginning of this lecture. So what does semi-conservative replication entail? It entails that the two strands of DNA separate and the new strands of DNA are formed on the two parental strands which have separated, right? The new strands of DNA form on the two parental strands which have separated out. See this sort of Y structure which you notice in this second part of the diagram right here. This is the process of replication taking place. And now we shall see how, right? We shall see how. So at the beginning, let's say this red thingy right here, I think I'll make it a more prominent red, right? Because the last diagram wasn't that very much prominent. Let's say this red thingy right here, this is my heavy DNA. It's heavy, right? All the nitrogenous bases are composed of 15 nitrogen or heavy nitrogen. So the entirety of this DNA is heavy, right? It's heavy DNA, right? And I have carried out CSCL density gradient on this one, and I have obtained this sort of a thing, right? All the DNA, it's heavy and at the same place, right? This is 15NH4Cl, right? And we also make a sort of plot diagram like this. Let's say this is one of the lines on my plot, this is the other line, heavy, intermediate, and this is light, right? And it will be entirely like this, right? Peak, because all of it is heavy, right? All of it is heavy, and this is CSCl tube, and this is plot, right? I'll actually CSCl, this is density plot, right? Density plot. Now this is transferred to 14NH4Cl, right? To 14NH4Cl. Now it's again subjected to density gradient after 19 minutes. 19 minutes, right? After 19 minutes. Now what happens? This is the really fun part right here. Since it's semi-conservative, you see one of the strands is the parental strand. This is the other parental strand. But now the daughter strand, this is the daughter strand, right? The daughter strand is different, and the daughter strand, it is derived from 14NH4Cl, right? It is derived from 14, the nitrogenous bases, the nitrogen in them is derived from 14NH4Cl, right? So it's a light strand, it's not a heavy strand as before, right? It's not a heavy strand as the parental strand. Now the two parental strands have separated, and the two daughter strands, they are both light strands, and so the DNA as a whole is intermediate in nature, right? The DNA as a whole is intermediate in nature. It's not heavy anymore, neither is it light, because the replication is semi-conservative, right? Because the replication is semi-conservative, one of the strands, the parental strand, is also there, and the daughter strand is also there. Since I'm sorry, the parental strands separate, and the daughter strands are actually formed on complementarily to the single separated strands of the parental DNA. So one of the strands is heavy, that is 15NH4Cl, and the other strand is light, that is 14NH4Cl, right? So this is a really cool inference, and this way actually this is the way in which DNA replicates, right? Now you can say, hang on a minute, we would also get the same density gradient graph. No, actually we wouldn't, right? Where we like you might say we will get the same density gradient graph if one of the DNA was entirely heavy and one was entirely light, right? No, we wouldn't, because the light DNA would be close to the mouth of the tube, and the heavy DNA would be close to the base of the tube and be visible as two distinct strands, right? As two distinct bands on the CSCl density gradient tube, right? But here we see only one band, right? We see only one band. If you remember the tubes I drew in the previous part, we see only one band, and it is somewhere right here, right? In the middle, between heavy and light, right? And we call that the intermediate band. Now if I draw the plot, this is heavy, intermediate, and light. You will see that it is like this. Single peak again, because all of it is intermediate, and it is intermediate. Yeah, that's just a tautology, right? Then again, after 19 minutes, and I'm just talking about one of the intermediate DNAs here, because it's actually irrelevant to draw out the fate of both the DNA molecules, because they are basically the same. They are both intermediate, so it's basically the same. Now what happens? This strand of the first molecule, it separates. Let's draw this strand. Okay, and this strand, this blue strand, it also separates. Okay, and I'll draw this blue strand like this right here. Okay, so I've drawn the two parental strands. Now what? Now the daughter strands, will they be red or will they be blue? They'll be blue red because it's still 14NH4Cl. It's still light nitrogen. So the daughter strands will also be light. And since I have depicted light nitrogen and light DNA strands in blue, I have to depict these daughter DNA strands also in blue, right? So both the daughter DNA strands will be blue, both the daughter DNA strands will be light, right? Yeah, both the daughter DNA strands will be light. Okay, okay. Now you can see where I'm going, right? And this is quite fun and quite genius, actually, isn't it? This is a really fun experiment and a real it it actually portrays sheer brilliance on the part of these two scientists. And I'll draw the tube. Now this time, as we discussed previously, there will be one intermediate tube that will be thinner than the previous one. This is the intermediate tube, and there will be one light tube, right? Both of the same thickness. You see, there's one intermediate DNA and one light DNA. So 50% intermediate DNA, 50% light DNA, and that gives rise to two lines of same thickness on the CSCl centrifugation tube, right? And now if I draw the plot, and this is heavy, you see the heavy DNA is being phased out, right? There's no more heavy DNA here because it's semi-conservative, right? If I draw the plot, there'll be one peak in light DNA, one peak in, and both the peaks will be of the same size. One peak in light DNA, one peak in intermediate DNA, right? Both the peaks of same size, signifying that the amount of intermediate DNA and the amount of light DNA is the same, right? And now we go to the final part of the experiment, right? Actually, they continued it for more generations, but we don't need to consider that. And in the final part bit of the experiment, I'll be drawing the fate of both these DNAs because both the DNAs are of different types, right? It's relevant now after 19 more minutes. Uh, this is one of the strands. This is one of the strands. Okay, and the other strand of this DNA is blue because it's light. Okay, then there is one more blue strand. Why is that? Because it's a second DNA that consists entirely of blue strands, and there's one more blue strand that is the second parental strand of the second DNA. And now the daughter strands, all of them will be blue, right? Why will all of them be blue? Because it's again 14NH4Cl, and 14NH4Cl gives light nitrogen, and that incorporates into the nitrogenous bases of the DNA strand, and the DNA strand subsequently produced is light in itself, right? So again, blue, 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 blue. Okay, and now just join the two strands like this. Just join the two strands like this. Okay, so what do you see now, right? There's three light DNAs and one heavy DNA. So 3/4 of the entire DNA is light and 1/4 is I'm sorry, I said heavy. I think it's not heavy. It's intermediate DNA. Okay, so 3/4 of the entire sample is light DNA and 1/4 is intermediate DNA. Now, can you guess why the line on the tube of the light DNA? Why do you think I said it was three times thicker than the line of the intermediate DNA? Because if you consider the ratio, light DNA to intermediate DNA, it's 3:1, right? It's 3:1. So science is all about logic. You read this experiment in your books as sort of a factual treatise, you won't understand anything because it's pretty arcane, pretty abstruse for someone who hasn't had exposure in this topic before, right? You won't be able to make out much by just reading unless the textbook is very good. Okay, so this is how we depict This is how it actually looks like in the CSCL density gradient tube in the third generation. And now I draw the plot again, heavy, intermediate, light, and now you see the light peak is 3 times taller than the, actually I went overboard with that, so just reining it back a little. You see, the light peak is 3 times taller than the intermediate peak, right? And this brings us to the end of the experiment, and this is how we infer that semi-conservative DNA replication takes place. Now, I told you that you would understand the significance of 19 minutes after I told you about the inferences, but you probably don't understand that till now, right? Yeah, I pulled a little trick on you. It's because I didn't tell you that the DNA that these guys were working on, it's still inside the organism. It's not like it's been taken out or something. It's actually an organism known as Escherichia coli, right? Let me put an asterisk. They worked on Escherichia coli or E. coli. We call it E. coli, but the entire name is Escherichia. It's quite a mouthful, coli, and be careful of the spelling. You can get it wrong quite easily. Escherichia coli, right? And Escherichia coli, E. coli, it actually undergoes one complete round of DNA replication in 19 minutes, right? So basically, the generations that they took, they were just subsequent generations of E. coli, right? So after 19 minutes, the entire E. coli DNA was replicated, and there were exactly double the DNA that was present in the previous generation. That's why they took it as they took the intervals as 19 minutes, right? And they worked on Escherichia coli. DNA replication time. DNA replication time equal to 19 minutes, right? Equal to 19 minutes, and that's why they took they took samples from the culture medium at regular intervals of 19 minutes, and they ran the CSCL density gradient accordingly, right? And so this was all about Messelson and Stahl experiment, and you can see by now, you can appreciate by now that it was a really ingenious experiment, and it proved the semi-conservative replication of DNA, right? It proved the semi-conservative replication of DNA. Just go through it once. I'm scrolling up once again. Messelson and Stahl experiment. Grew DNA for several generations, conducted CSCL density gradient, transferred to NH4C, 14 NH4Cl, and another CSCL, 19 minutes. The time is important. 19 minutes, 19 minutes, 19 minutes each time. So 19, then total 38, then total 57, like that. And they continued for several generations, but I've only shown you 3 generations in this one. And the inferences are the most important part, and it can be inferred. This is how actually they inferred that semi-conservative DNA replication was taking place, by drawing the density plot and by seeing the CSCL, the patterns on the CSCL tube, right? Okay, so if the method of replication was anything other than semi-conservative, the patterns on the CSCL tube and the plots would have been different, right? I'm sure you can gauge, like if it was conservative, that is, the entire parental strand was conserved, and there was another DNA strand, another daughter strand that was produced, that was suppose entirely blue in color, means entirely out of 14 NH4Cl, then you would have seen 2 bands on the density plot, which would have been on one on the light and one on the heavy side, and both of them would have been in equal amounts, so the 2 peaks would have been seen, right? But that's not what we see. We see one intermediate, right? And particularly the fact that the heavy DNA is entirely removed after a few generations, after actually one generation, that is perhaps the most striking factor, right? Perhaps the most striking factor. Okay, now we've just let me add as a sort of addendum that this was done on prokaryotes, right? Escherichia coli is a variety of, is a species of bacteria, not variety. Variety has a different significance when it comes to biology. It's a different category altogether. It's a species of bacteria, and bacteria are prokaryotes, right? And this experiment was also carried out on a eukaryotic species known as Vicia faba. It's a species of bean. Faba beans? Have you ever eaten faba beans? You may or may not have eaten them, but just know for a fact that they are a species of beans. So faba beans, Vicia faba, this experiment was carried out on Vicia faba by Taylor et al. Taylor et al. Et al. Means Taylor and his team, basically, right? Taylor was the leading scientist in that team, and et al. Refers to the other members of his team. So Taylor and his colleagues, they carried out a similar experiment on Vicia faba and also proved that DNA replication takes place uh, uh, semi uh, semi conservatively in eukaryotes, right? Okay, now let's move on to DNA replication, process of DNA replication, right? Process of DNA replication. Right. So, about the process of DNA replication, the first thing that I will be talking about is the enzymes, right? Enzymes. Now, this is probably the most important bit, and I hope you know what enzymes mean. Enzymes are nothing but uh, biocatalysts, right? You know what a catalyst is? It speeds up a chemical reaction. Now, it may be an inorganic catalyst. Say, if you've heard of the Haber process, iron is used as a catalyst, right? Iron is used as a catalyst, and what it does, it speeds up the rate of the transformation of nitrogen and hydrogen to NH3 to ammonia, right? Or maybe some other process like the Ostwald process. Platinum is used as a catalyst, or platinum rhodium gauze is used as a catalyst, or in the contact process, vanadium pentoxide is used as a catalyst. If you don't know these processes, it doesn't matter much, right? I'm just giving some examples off the top of my head, but if you don't know these processes, it's no matter. I'm just giving them as examples uh, of catalysts, right? Of catalysts. Catalysts, basically what they do is speed up a chemical reaction. Now, enzymes, they are a special class of biocatalysts. They are mostly proteinaceous molecules that speed up chemical reactions in the living body. And the most important and primary difference with other inorganic catalysts, which I was talking about earlier, is that the rate by which they increase the speed of these chemical reactions is of these biochemical reactions is much more than the inorganic catalyst can even think of, right? Like if you consider a basic metabolic reaction like respiration, if you consider combustion and respiration are basically similar, right? By addition of oxygen, uh, basically both involve addition of oxygen and release of energy, right? That is the basic thing that's similar in both combustion as well as respiration. So if you consider combustion, that occurs at very high temperatures, right? But if you consider respiration, it's 37 degrees Celsius, our body temperature. In organisms that cannot regulate their body temperature, it occurs at even lower or higher temperatures, depending on the ambient temperature, the surroundings around them, right? So the energy barrier, the activation energy, so to say, if you don't know what activation energy is, it's no matter. But basically, see, and when a reaction takes place, there's an energy barrier that needs to be overcome, which is known as the activation energy, right? So what the enzymes do is they lower that activation energy, so the reactants to products, the transformation of reactants to products can take place at a faster rate, because so that they can easily jump the energy barrier, right? So in combustion, let's go back to that example. In combustion, that energy barrier is pretty high, right? And we need to supply that initial heat energy in the form of a high temperature in order to make the molecules undergoing combustion to jump the energy barrier, to overcome the energy barrier, right? But when it is catalyzed by an enzyme, the energy barrier is so low that even at 37 degrees Celsius, the molecules have no problem jumping that energy barrier and going right to the products and releasing energy, right? Okay, so that is basically the role of an enzyme, right? I'll make a video on biomolecules pretty soon, and I'll delve into these topics in even more detail, so don't worry, right? So enzyme, the first enzyme I want to talk about is DNA dependent DNA polymerase. Now we are talking all about prokaryotes, right? Because in eukaryotes it's much more complicated, and we don't really need to delve into that at this level right now. Okay, this is a grade 12 level AP bio level, O level biology course lectures, and even if you're preparing for Olympiads, this is all that is required. You don't need to know much about eukaryotic replication, right? DNA the first enzyme I want to talk about is DNA dependent DNA polymerase, right? DNA dependent DNA polymerase. And in prokaryotes, it's of three types: DNA polymerase 1. I'm just referring to it as Pol, DNA Pol 1, DNA Pol 2, and DNA Pol 3, right? DNA Pol 3. Now, polymerase, what does that name kind of tell you? It's similar to polymerize, right? Polymerize, and since it's an enzyme, that A's term will always be there at the end. If you've heard of other enzymes like, say, maltase, sucrase, lactase, amylase, that term is right there at the end, right? So polymerase. So polymerase is an enzyme that polymerizes, right? Polymerizes. And what does it polymerize? It polymerizes DNA. And what are the monomers of DNA? If you know what a polymer is composed of, a polymer is composed of monomers, right? What are the monomers of DNA? The monomers of DNA are nucleotides, right? We learned about that in the previous lecture, in the previous video. The monomers of DNA are nucleotides, right? So DNA polymerase polymerizes the monomers of DNA, that is the nucleotides, into the nucleic acid strand, right? Into the nucleic acid strand, the DNA strand, right? Okay, so this was about DNA dependent DNA polymerase. Now let's talk about another enzyme called helicase. I'll tell you the functions of all these enzymes later. Let me tell you about them right now. There's nothing to it that's too obscure, right? So helicase, what it does is it opens the two strands of DNA since they are in a helix, right? A helicase, you kind of gauge that from the names itself. See, don't try to remember the names like that. Just try to connect the name to the function it performs, right? Helicase, you see the helix is right there at the beginning of the name, right? So helicase, what it does, it unwinds the helix, right? It unwinds the helix, it opens the helix. Say, suppose the helix is like this, so the helicase makes it like this, right? It opens one part, the remaining part is still a helix, but it opens one part so that replication can proceed in that part, right? So that replication can proceed in that part. So helicase opens the helix of DNA. Now let's talk about topoisomerase. 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 Topoisomerase, basically what it does, if you have you ever twisted two pieces of string on themselves? Have you ever, like, sort of, you twist two pieces of string on themselves and you let them go, and then suppose it's a bag or something beneath it, and then it twists and returns to its original position, something like that. Have you ever done that in your childhood, or something akin to that? Well, see, basically, or have you seen nylon rope? A nylon rope is something like this, right? It contains many strands that are coiled around each other like this, right? So basically, when you open a part of the strand, the end that is away from it, it immediately gets super coiled, right? It becomes much tighter. If you have ever done this with two pieces of string, you'll notice that the remaining portion of the string, the coiling on the remaining portion of the string, it becomes much tighter, right? And it gets knotted and it gets entangled and all that sort of stuff. We can't let that happen to our DNA, right? We can't let that happen to our DNA, right? Otherwise we are in a bucket load of trouble, right? We cannot let that happen to our DNA. We are in really hot water if that happens to our DNA, right? So topoisomerase, what it does, it basically relieves the tension. It relieves the supercoiling tension. We call it the torsion of the remaining part of the DNA, right? The torsion of the part of DNA that is still helically coiled, that hasn't been opened by helicase yet, right? So topoisomerase, it's a very important enzyme. It relieves the tension, the tension of supercoiling in the remaining part of DNA that has not been opened by the helicase enzyme, right? That is the function of topoisomerase. Okay, what enzymes do I have left? Let's talk about RNA primase, or simply primase, you may find written in some books. Now, the thing about DNA polymerase is that if it does not contain some nucleotide to guide it on its way to polymerizing, it cannot polymerize, right? DNA, it is incapable of polymerizing without a primer. We call it a primer. See, primase, the first word, the first part of the word, it's akin to primer, right? It's prime. Prime to primer isn't a long shot, right? So RNA primase, it basically sets down an RNA primer on both the open strands of DNA, so that the subsequent DNA replication via DNA polymerase, via DNA dependent DNA polymerase, can take place smoothly, right? Because DNA dependent DNA polymerase requires an RNA primer to begin its process of polymerization, right? This is the function of RNA polymerase. And finally, I have another enzyme called DNA ligase, right? DNA ligase, and its function is to join the discontinuous strands of DNA which are known as Okazaki fragments. But I won't be able to explain it to you in detail if I don't move on to the next part of this lesson, right? So just know that DNA ligase is also one of the important enzymes, and I'll tell you about the function when I come to the exact process of what occurs, right? Okay, now I'll just pause this video, this lecture for a moment, pause the recording for a moment actually, to draw a diagram. If I draw it while the video is still recording, it will take a lot of time, right? And it'll make the duration of this video really long. So just to cut down on that a little, I'll just pause the recording for a while. You won't notice anything, okay? Okay, so I have drawn the diagram, and let's proceed with the labeling now, right? Let's proceed with the labeling. It's just a bunch of squiggly lines and colors for now, but I haven't labeled it on purpose. This big yellow thing that you see right here, this is what is the topoisomerase. You see this, it is attached to the part of DNA that is still helically coiled, right? It is attached to the part of DNA that is still helically coiled, so it has got to be the one that's relieving the supercoiling, right? So this is the topoisomerase enzyme. This is one of the strands of DNA, this blue one, and this is another strand of DNA. It's pretty evident. This triangular sort of thing right here is known as helicase or helicase, right? This is opening the helix of the DNA, and you see the two strands are nice and separated now, right? The two strands are nice and separated. Now this yellowish orange bit right here, what do you think this is? Uh, I haven't told you about this. That's because it's not an enzyme, right? It's not an enzyme. This is known as single-stranded binding proteins. Single-stranded binding proteins, also known as SSBP, right? Single stranded binding proteins, also known as SSBP. Okay, if you watch the thumbnail of the video closely, you will notice that I've written the abbreviation there, SSBP. But if you didn't know what SSBP stands for, know it now. It stands for single stranded binding proteins. And what they do, see, DNA in its natural state, it's helically coiled, right? So if it's been opened, it wants to go back into that helix, right? It wants, because you already know, if you watched the previous lecture, that there are hydrogen bonds, there are complementary hydrogen bonds between the purine and pyrimidine bases on the two strands of the DNA, right? So they want to, the hydrogen bonds, they are attractive, and they want to go back to that initial configuration of a helix coil, right? So once it has been opened, there needs to be something that stabilizes it in that open state, because that open state is less stable than its natural helical state, right? So the SSBPs, they are actually involved in the stabilization of the in the stabilization of the separated DNA strands, right? They are involved in the stabilization of the separated DNA strands, and that's why single-stranded binding proteins are important. Now, if you know the convention for numbering the two DNA strands on the basis of directionality, I have talked about it in the previous video. Please go back and watch it if you haven't, because it's very important to understanding this one, right? Otherwise, you'll be lost in the water. Okay. So this is the 5' strand, let's say. So this automatically becomes the 3' strand, right? So 5' and 3', right? 5' and 3'. Okay, now we can proceed. Now let's say I have, before that, the first step which takes place. This step It takes place in the first step that takes place in the nucleoplasm or the cytoplasm, mostly in the nucleoplasm if it's a eukaryote. But since we're talking about prokaryotes, it's the same as the cytoplasm. I'm sorry, scratch out the nucleoplasm part. It takes place in the cytoplasm. What happens is that nucleotide, or rather nucleoside monophosphates, you're kind of confused, right? Nucleoside monophosphates, why not just call them nucleotides? A phosphate group attached to a nucleoside is just a nucleotide, right? Well, there's a reason why I'm referring it to it in this way, are converted to nucleoside triphosphates. Are converted to nucleoside triphosphates. Nucleoside triphosphates. This is done by a special category of phosphatase enzymes, right? Phosphatase enzymes. Nucleoside triphosphates. Now, why is it done? Have you ever heard of something known as ATP, adenosine triphosphate? What is that? That is a nucleoside triphosphate, right? If you don't get it, let's see. Let's say this is adenine, which is attached to my ribose, right? This is deoxyribose in this case, right? This is should be deoxynucleoside monophosphate, but that is a venial error. Let's say this is HOH2C, right? And this is a phosphate group. Okay. Okay. So this is a nucleoside monophosphate, and this is adenosine monophosphate, AMP, right? Have you heard of ATP? What do you think ATP is? Adenosine triphosphate, right? Now ATP is the energy currency of any living cell, right? So where is that energy stored? Well, it's actually stored in the phosphate bonds. Once you have more than one phosphate bond, the other phosphate bonds are all high energy phosphate bonds. They are high energy phosphate bonds, and as a result, the molecule that is thus formed, or adenosine triphosphate, that once you break those phosphate bonds, you can get a lot of energy from that, right? And that is how energy barter takes place within the cell. When when a metabolic process releases energy, we form those high energy phosphate bonds so that it can be stored, and wherever that energy is required, the ATP can travel to that place and it can break those bonds, and the energy is released for the process to take place, right? So high energy phosphate bonds, if I was to depict, I'm sorry, yeah, if I was to depict the high energy phosphate bonds, they would be kind of like this. More phosphates, uh, more phosphates, there's one more phosphate, and this is adenosine triphosphate, right? This is adenosine triphosphate. Okay, so these bonds, they are high energy bonds, right? These are high energy bonds, and once they are broken, they release a lot of energy. And why do we require that energy? We require that energy because DNA polymerization, it is an endergonic process, right? DNA polymerization, since we are forming something, it is an endergonic process, and so the energy that is released by the breaking of these bonds, by the breaking of the nucleoside triphosphate into the nucleotides that are actually required in the DNA, that is the nucleoside monophosphates, that energy is required in the polymerization process to join the adjacent nucleotides together, right? That's why nucleoside monophosphates are converted into nucleoside triphosphates, right? Now, after the nucleoside monophosphates have been converted to nucleoside triphosphates, the not the helicase unwinds DNA at a special location known as the origin of replication, known as the origin of replication, right? Known as the origin of replication, and we call it ORI, ORI, or ORIC, or ORIC. You can call it ORI or you can call it ORIC in prokaryotes, right? Helicase unwinds DNA. So in this diagram, this is the origin of replication, right? This is the origin of replication, and helicase unwinds the DNA at this spot, right? Now after that, primase, mm, primase, what do I say? Primase, let's say, sets down, right? Sets down complementary RNA primer, complementary RNA primer, a primer, right? So the primase, it's a big enzyme, it's an enzyme, it comes one primase like this on this strand and another primase like this on this strand, and it sets down the RNA primer, right? Let's say, let's make it yellow. Let's say this is one of the RNA primers, this is one of the RNA primers, and this is the other RNA primer, right? The other RNA primer, and it's complementary to the strand, right? So it's complementary to the strand. Let's say if I have a sequence like AAT in here, this will be TAA, or if I have a strand like GAT in here, it will be like CTA. All right, so it's complementary, right? It's complementary, and it binds to the two DNA strands. Now you must be wondering, why have I drawn one of the RNA primers at the fork of the fork where the helicase has opened the DNA, and the other at the end, right? That is because the polymerization of DNA can only take place in the 5 prime to 3 prime direction, right? 5 prime to 3 prime direction. Know that for a fact. Know that for a fact. That is a fact. My lectures have very few facts, but the ones that they do have, they are very, very important, right? DNA polymerization via DNA dependent DNA polymerase, specifically via DNA dependent DNA polymerase 3, can only take place in the 5 prime to 3 prime direction, right? So, but how does that figure into here? Now let's see. This is the 3 prime end, right? So for the RNA primer that's been set at this end, this will be the 5 prime end. Since it's complementary, right? It's the complementary end, right? So this will be 5 prime, this will be 3 prime. And now the DNA that will be further polymerized, it will start from the 5 prime end, since the 3 prime has been occupied by the end of the RNA primer, right? So it can proceed in this direction, right? 5 prime to 3 prime direction like this, 5 prime to 3 prime direction like this. So it can proceed without any trouble, right? But if the, I'll rub this bit out. But if we had set down the RNA primer right here, what do you think would have happened? This would have been the 3 prime end, this would have been the 5 prime end. Now when the DNA polymerase comes in and wants to polymerize, this is the 3 prime end, this is the 5 prime end, right? Oh my god, it cannot polymerize. It cannot polymerize. It can only polymerize in the 5 prime to 3 prime direction, right? It has got a sort of liability, a sort of disability in this way, right? That it cannot polymerize in 3 prime to 5 prime direction. It can only polymerize in 5 prime to 3 prime direction. So in order for that to happen, we need to make sure that the RNA primer is set properly, right? So in case of this strand, since this is the 5 prime end, this has to be the 3 prime end, right? This has to be the 3 prime end, and so this end will be the 5 prime end for the RNA primer, and this will be the 3 prime end for the RNA primer. So now this will be 5 prime for this for the daughter strand laid down by polymerase, and this can now go in the 5 prime to 3 prime direction. And this is why one of the RNA primers is set on the towards the fork side, and the other RNA primer is set towards the end of the strand, right? Now comes the boss, the main guy of the party, and he is, or she is, whatever you desire, the DNA dependent DNA polymerase 3. Okay, DNA dependent DNA polymerase 3. That is very important to remember, that it is DNA dependent DNA polymerase 3, right? It is the main enzyme that actually polymerizes the DNA, right? DNA dependent DNA polymerase 3 on both sides. And what it does, let me change the color to something of a, let's see, let's make it, I think green would look nice, but it would interfere with the helicase, right? Let's make it a light lavender color, right? And now it polymerizes DNA like this. It actually travels along the DNA. It's initially, let's say it's at this point, Let me make it dotted so that you can understand how it works. Initially it's here, then it moves here, then it moves here. You understand what I mean? It's actually, it's moving along the DNA strand, right? It's moving along the DNA strand, and it's polymerizing the DNA strand like that, right? So the DNA pol 3, it's actually polymerizing in that way, right? And it's polymerizing in this direction, right? It's polymerizing in this direction, and the the DNA pol 3 in this, on this side, is polymerizing in this direction, is polymerizing in this direction, right? Now you may be wondering, if you are inquisitive, why did I draw one single straight line for the end where the RNA polymerase is attached to the end of the, for the strand on which the RNA polymerase, the RNA primer is attached to the end of the strand, and why did I draw dotted arrows? I mean, number of arrows on the strand in which the RNA primer is attached towards the fork side. Well, that is because of another really nice, really cool phenomenon known as semi-discontinuous replication. I know this can get a little bit confusing if you are doing this for the first time, right? It's got such a lot of intricacies, such a lot of nitty-gritties, a lot of details that you feel like it's all over the place, right? It's a very intricate process. It has to be, right? Because it's about life and death. Life would not exist if DNA did not know how to replicate, right? Life would not exist as we know it, because this is this is the um, this is, what do I say? This is the deepest zenith as well as the absolute azimuth of life, right? The absolute crux of life as well as its deepest abyss as well, right? Because it's the most basic thing, the core from which all life originates, but it's also the pinnacle of the evolution of life as we know it, right? So it has to have a lot of checks, safeguards, uh, a lot of nitty gritties, a lot of details, right? A lot of intricacies. So why does this semi-discontinuous thing happen? Let's see. Suppose I have a little bit of DNA opened like this, and the rest of it is helically coiled, right? Helicase is open. Now I'll make that diagram a bit bigger. Let's say I have just opened that much of DNA, right? Okay, and on this end it's fine. It's formed a long strand like this. Okay, and there is a bit at the end which is a primer. Okay, but on this end the primer is on this side, and this is the strand which has been formed by the polymerase, right? This is the strand. Okay, now I have already replicated what was there to replicate on this amount of DNA that's just been opened, right? So now a little more of the DNA has to be opened. Let's say a little more has been opened like this. Okay, a little more has been opened. Now what? Now this strand, since this is the 5 prime to 3 prime direction, this can keep on replicating as it is, and it can reach to the end, right? It can reach till the end, right? It has no problem. It's fine with everything. It's a cool dude, yeah? It can just go on as it was. But this guy, it needs a new primer here, right, towards the fork side, because it's going in the 5 prime to 3 prime direction. It's kind of disabled in that way, right? So this strand has already been formed, right? This strand has already been formed, but it's it begins from kind of about the center of the DNA, of the parental DNA strand in this diagram, right? Because it's proceeding in the 5 prime to 3 prime direction, it's got a handicap in that way. So the other primer must be set at the fork again, right? The other primer must be set at the fork again, and now the DNA polymerase needs to polymerize another strand like this, right? It needs to polymerize another strand like this, right? So it polymerizes another strand like this, but you see it's not continuous, right? It's not continuous. It's got a gap in the middle right there. It's got a gap in between the two strands right there, and that is why this strand is discontinuous, right? The replication on this strand is actually discontinuous, right? Because you have got one daughter strand like this, that's fine. Then when it unravels, when it unwinds a little bit more, you've got another daughter strand um, that's from behind it, from the fork end towards the free end, but the two are not connected. Whereas on the other hand, on the other strand, you see the entire strand is continuous, right? The entire strand is continuous because it's proceeding in the 5 prime to 3 prime direction towards the fork, and it has no need to set another primer because the initial one works just fine. The DNA polymerase can go on polymerizing, right? And this strand therefore is known as the I think I'll just erase these makeshift diagrams and do it on the real thing. Let's do it on the real thing. And this strand, what color should I use? I should use this light pink. This strand, therefore, is known as the lagging strand, right? The lagging strand because it lags. It lags behind in replication because you see another primer has to be set. Another DNA polymerase has to come in and has to make that sort of a little bit of daughter strand, and then it actually lags behind this other guy right here who can just breeze through with one polymerase and one primer and can just continuously create a strand. It's like one guy who is laying down an asphalt road and another one who's laying the rail tracks, right? The rail tracks have to be laid down one by one, takes a lot of time. But asphalt road, you just dump all the tar on top of whatever bricks or concrete you've got at the bottom, and you can be done with it, right? So it takes a lot less time than actually laying down the rail tracks, right? The lagging strand, it's like the guy who lays down the rail tracks, and this other strand, it's known as the leading strand because it leads, right? It's faster. It's not exactly faster. We won't call it faster, but um, we humans kind of have a tendency to humanize everything, right? This guy is leading because he's running through, breezing through without any need for any other primer, without any need for any other polymerase. Whereas this guy, it needs a new primer each time, it needs a new polymerase each time, and then it just grows in spurts, right? It grows and then it stops, it grows and then it stops, right? So that's why this guy is called the lagging strand, and this other guy is called the leading strand, right? And these little fragments of DNA, of daughter DNA, that are formed, these are known as Okazaki fragments. Okazaki fragments. Okazaki was the name of a scientist and it's named in his honor. So these are known as Okazaki fragments. Fragments, not enough space, but let's just do it right there. Yeah, Okazaki fragments or Okazaki filaments, right? Now, let's say the entire DNA has been replicated in this way. Everything's fine, everything's great. DNA is very happy now that it has got kids, right? It's got kids. The DNA has got kids, right? Now what happens? Now what happens? Now the Okazaki fragments, they need to be joined, right? The Okazaki fragment. First, the primer needs to be removed. Why? The primer is composed of RNA, right? The primer is composed of RNA. Now you may think, what's the difference? Whoa, how does it matter? Well, there's a lot of difference. First of all, the sugar is different. The sugar is ribose and not deoxyribose. DNA cannot function with ribose, it needs deoxyribose, right? So the sugar is different. Then RNA contains one nitrogenous base, one pyrimidine that's different from that present in DNA, right? And RNA contains uracil, whereas DNA contains thymine, right? Thymine is a pyrimidine, uracil is a pyrimidine that is present in RNA. Thymine is a methylated uracil. It's a methylated form of uracil only, but it's different, right? It may be methylated, it may just differ by a methyl group, or it may differ immensely, but it's different, right? So thymine is a methylated uracil that is present in DNA. So since the two pyrimidines are different, DNA and RNA, the RNA primer needs to be removed, and a short DNA strand needs to be put in its place, right? A short DNA strand that is the size of the RNA primer needs to be put in its place, right? So what do you think? Which enzyme do you think does this, right? This is done by DNA polymerase 1, right? I'll just rub out the DNA polymerase 3s because since I'm not really, since that step is over, I am interested, but that step is over. So I'm removing that enzyme, right? And I'll just put the bonds in between the two strands. I put the bonds in between the two strands to signify that these are DNA strands. And now I have got my next enzyme, that is DNA pol 1, right? DNA pol 1, let me signify it in that way. This is DNA polymerase 1, right? So what the DNA polymerase 1 does is it First of all, it has exonuclease activity, right? It has exonuclease activity, and so it removes the primer, and it also has polymerase activity. It is, after all, a polymerase, right? So it removes the primer and it sets down DNA in its place, complementary DNA in its place, right? This primer is also removed and complementary DNA is put in its place, right? Okay, actually, I made a little mistake there. It's still separate. Okay, it's still separate. Okay, and there are primers in between each of these Okazaki fragments as well, which I have not shown, but actually they are there because every time a new round of opening of the two strands of DNA and setting down of the primers is taking place, right? So each of these Okazaki fragments has an RNA primer at the end, and what the DNA Pol 1 does is that it removes the primer and it sets down the complementary DNA, the DNA, the daughter DNA strand that is complementary to the parent strand, right? So that is the function of DNA Pol 1. Okay, now we have the entire, all the kids of DNA are DNA only. DNA has no RNA kids anymore, right? DNA has no RNA kids anymore. All the kids of DNA are DNA only. And now there's another there's another slight dilemma, right? It's full of slight dilemmas like this one, right? The entire process, you may see, it's quite tedious to actually go through. It's quite tedious to actually go through the first time, but if you enjoy it, if you try to visualize it, you see that it becomes, it's very interesting. You can look at it in two ways. Basically, you can look at it as either a tedious succession of events, or you can look at it as an interesting process that is the root of all life itself, right? So now you see there are many gaps between these. There are many gaps between these Okazaki fragments. There is one gap between the primer and between where the primer was, the erstwhile primer was, and the rest of the polymerized DNA strand. So there are gaps, right? And these gaps need to be filled. Gaps never do good, right? You dig a hole and you fall in it yourself, right? So gaps are never good. Everything needs to be continuous. Everything needs to be smooth. So that's why we have something. You remember DNA ligase? Do you remember DNA ligase? I told you that I could not explain its function without referring to this diagram, right? This is why. Okay. So what DNA ligase does is it basically joins these gaps. It just joins these gaps, right? It just joins these gaps, and this function is done by DNA ligase. Let's say this elongated sort of thing is DNA ligase, and its basic function is to join these gaps so that we have two continuous strands of DNA, right? Both the lagging strand and the leading strand are continuous, right? So the semi-discontinuity, that is the discontinuity in the lagging strand and the continuity in the leading strand, both of them become continuous. Both of them become, what do I say? Both of them become coherent in a way, and that is the function, the final function of the DNA ligase, and that that isn't the end of uh, the replication process. There's still one more tiny bit. There's still one more tiny bit. I know this video is getting pretty long, but the you can't help it. It's DNA replication, right? It's the basis of life itself, right? So there's one more process, that is proofreading, right? Proofreading. You see, I have pretty much explained all the jobs of all the enzymes right here, right? Right, the functions of all the enzymes right here, right, through this diagram. But DNA Pol 2, what does it do? DNA Pol 2, it hasn't come up anywhere in this entire process. It hasn't come up anywhere in this entire process, right? What does DNA Pol 2 do? Well, DNA Pol 2 is the enzyme that is involved in the proofreading process. Now, what is proofreading? Now, suppose you have polymerized a lot of DNA, and say you have a DNA strand that goes adenine, adenine, thymine, guanine. So it should be thymine, thymine, adenine, cytosine, right? It should be thymine, thymine, adenine, cytosine, right? But suppose in place of the adenine, you have placed a guanine. So guanine and adenine are not complementary bases, but it happens sometimes. Can be a purine in place of a purine, or a pyrimidine in place of a pyrimidine. This is the basis of mutation, right? If a different purine is placed in place of one purine, the process is known as transition, and if a pyrimidine is placed in place of a purine, the process is known as transversion, right? So in case that sort of thing happens, let's say in place of adenine, guanine has been placed by mistake, right? Mistakes do happen, mutations do happen. Mutations actually may be beneficial as well as harmful. It may form the basis of evolution, or it may lead to the death of an individual. Even it may lead to more an individual that is more evolved to meet its needs, or it may also result in an individual that dies very shortly after birth or before birth itself, right? So mistakes do happen, and these mistakes, they are corrected by DNA polymerase 2. That's why more mistakes don't happen, right? Mistakes do happen by the DNA polymerase 3, right? So to correct those mistakes, let's say there's one mistake every 1,000 base pairs. If there's one mistake every 1,000 base pairs, there'll be a million mistakes, and every kid that will be born will have some defect or will be a superhuman, right? So we cannot really afford that. As a species, humans or any other species for that matter can't really afford that. So we have DNA. We don't have prokaryotes have. We have an entirely different system altogether. We have alpha, beta, gamma, delta, epsilon, different types of polymerases. But the prokaryotes, they have DNA polymerase 2, whose function is to proofread and to weed out these mistakes committed during the DNA replication, right? To weed out these small mutations, these small point mutations that may have arisen. Uh, Even the DNA polymerase 2 is not infallible, is not foolproof, because mutations do take place even after the proofreading process, but they are much less in number because the DNA polymerase 2 is there, right? So that was actually the last bit of the replication process that you needed to know. And this lecture has gone on for 1 hour and 30 minutes. I all my lectures are quite long, mostly in excess of 1 hour, but this is a little bit uh, longer than my previous ones, but it's justified because this is DNA replication. We are talking about a very important process, and we have come to the end of our lecture for today, and we can begin genetics, I feel, in my next lecture. So bye for now. Have a great day and see you in the next video. Thank you.